So why don't we get started? Today we're very pleased to have both teams involved with PANC-DB and also PanKbase to give an overview of their resources. And so first off, we are going to hear about PANC-DB, and Dr. Robert Faryabi will, will be able to introduce his team, and then we'll get along with that presentation. And then after that, Dr. Marcela Brasova will introduce her team and will discuss PanKbase. So why don't I turn it over to you, Dr. Faryabi, and
Thank you.
Nilanjana, if you need to share, just let's make sure you can share your screen.
Okay.
Okay. And Dr. Faryabi, did you want to give a quick introduction?
Just very brief introduction. First of all, thank you for this opportunity. We are excited to share some of the work we have done in the last, I should say, 7, 8 years. We are part of a program called Human Pancreatic Analysis Program that many of you on the call might be a member of. Unfortunately, I can't see the list of all the attendees, but we are mandated by generating a deep molecular phenotyping of pancreatic tissues and related immune systems that involves in type 1 diabetes pathogenesis. So PANC-DB shares this data with the community. We provide some functionalities, and some of our data lives on PanKbase, as you will hear in the second half of today's discussion. With that, I would like to hand into Nilanjana, who kindly agreed to make the presentation while attending a wedding. Thanks, Nilanjana.
Okay, great. So good afternoon, everyone. I am Nilanjana Samanta. I manage the pancreatic database, or PANC-DB as it is commonly known as. It is a free, publicly accessible data repository, data dissemination, visualization, and analysis platform for all cellular and molecular data sets generated by the Human Pancreas Analysis Program, or HPAP, just like Dr. Babak Faryabi just mentioned. So this is a brief outline of my presentation today. I'll attempt to keep it precise, but I apologize in advance if I go over my 20-minute time. It just means less questions for me. So I'll give you a brief overview of PANC-DB, briefly go over the data, different types of data that we currently host, and then finally give you a live demonstration using a use case example. So if you are interested in coming along with me as I give the live demonstration, it's fairly easy to search for our database. Just search with the letters like shown here, PANC-DB, or use this URL shown here and paste it on your browser, and we should be among your top 3 hit. So what you see here is the operational workflow of the HPAP program, and PANC-DB is the public-facing entity of this program. We have an array of different kinds of data sets, like imaging data, genomic, epigenomic, transcriptomic, islet function data, as well as immune profiling data for all of our donor samples, which makes us extremely unique because it allows us to give a multiplexed view of the disease. All of these data are generated not just from pancreatic tissue and islets, but the immune profiling data comes from peripancreatic lymph node and spleen, in addition to the more traditional method of using peripheral blood. So all of this together makes us the largest database for original multimodal data for both type 1 and type 2 diabetes, as well as autoantibody positive donors, which I'll explain soon. We also attempt to harmonize and integrate our data with related resources in this research space. For example, our imaging data is available for exploration on PanKbase. And like Dr. Babak Faryabi said, later in today, you will see that our data set is also available to explore via the various tools that PanKbase has on their platform. And our data is also available on various other platforms as shown here. So now I'll quickly go over the different kinds of data that we host on PANC-DB. But before I do that, I'd like to mention that HPAP is a multi-institutional effort comprising of experts from all of these different institutions who contribute to the data generation effort. So as of today, we host data from a total of 193 donors that are live on PanC-DB. And within the next 2-3 weeks, when I'm back in office, you will see data from at least 200 more donors. Um, and all of these donors can be grouped into these different disease categories, that is type 1 and type 2 diabetes, autoantibody positive donors, and these non-diabetic controls. We, in addition to the type 1 and type 2 diabetes cases, we also collect age-matched controls, which are these non-diabetic controls. So these non-diabetic donors can be further grouped into a subtype called the autoantibody positive subtype. These are donors that did not have a clinical diagnosis for type 1 diabetes before they passed. However, they were found to be positive for one or more combination of the 4 different kinds of beta cell autoantigen that we know of that are crucial for type 1 diabetes detection. So as you can see, this is a very valuable data set for our researchers. Here you're looking at a comprehensive list of different assays for which we host data. The ones highlighted in red are the ones that we recently added to our database, which includes Codex imaging, both dual and triple modality single cell multiome data for immune profiling. And for our islets, we just added the single nucleus RNA-seq data. All of these allow you to do in-depth characterization of the diabetic tissue. We also perform whole genome sequencing on all of our donor samples. However, that data is available only on dbGaP for HIPAA concerns. On PANC-DB, although you can see the information on its data availability, where you can see which donors have WGS data on dbGaP available versus those who do not. Now, the counts on the right-hand side of these tables are nothing but the donor count, meaning donors with at least one data set available for all of these different assays. And you can see that they are not all at 100%. That's because we attempt to conduct this entire panel of experiments on all of our donors. However, it highly depends on the quality and the quantity of the islets or the tissue or the sample procured for each donor. It is also important to keep in mind that ours is a dynamic database, meaning donor identification, sample processing, data generation, and data upload are all happening simultaneously at different timelines in real time. So this can lead to differences in data availability. And during my live demonstration, I'll show you exactly how we attempt to communicate that to our users. So now it's time for the live demonstration, where I will use a practical example and attempt to show you the utility of not just our interactive tools that are basically decision-making tools for our users, but I also want to show you how you can query our database, make a study cohort, and download the raw data. So the use case that I will be using today is this one. According to studies, we know that GATA-positive individuals are highly likely to develop type 1 diabetes. However, not all GATA-positive individuals end up developing type 1 diabetes. So now let's start with the live demonstration. And in the interest of time, what I've done is I've already opened different tabs for different windows on our database, just in case we cannot trust the internet speed up in the mountains where I am. So this, once you search for the PanC-DB database, this is the home page you will land on. And right off the bat, you can see these 4 major tabs. These are the primary data exploration options that we provide. I will go over them one by one, but before I do that, I want you to look at this top navigation bar, because this is the navigation bar that will stay with you as you open different windows on our data on this website, and move away from the home page. So these 4 major tabs are also available in this top drop-down menu over here. If you're interested in watching in-depth video tutorials on how to use PanC-DB, download data, and use its various interactive analysis tools, they are all available on this tutorial page, along with GitHub pages to various tools that our team has developed. If you're interested in downloading these standardized protocols for the various assays I listed earlier, you can do that on this workflow and protocol page. Just navigate to the assay you're interested in, and then you can download the PDF for that assay or just read it over here on the website. Clicking on this logo right over here will allow you to come back to the home page. And before I exit this page, I wanted to show you these 2 summary stats because they're pretty useful. So on the right over here, you're looking at a plot that's showing you the availability for all of these different assays listed on the y-axis for a donor by donor count. And they are ordered by most abundantly available on PanC-DB to less abundant. And they are divided into different disease groups. And on the left here, you can look at the number of publications over the years that have used PanC-DB data. The 2025 data is only up to September of 2025, and it's bound to go up. Let's look at how you can query our PanC-DB and create a study cohort and look at different clinical profiles of the donors. So for that, I have come to this donor information page, which is also available here on this drop-down. On this page, I did not mention to you earlier that in addition to the data for all of the various assays I listed, we collect in-depth clinical and demographic information on all of our donors, and which you can download using this tab over here for all of our donors. Out of all of those clinical and medical history information, we've selected some key medical information and demographic features and use them to create this filter on the left-hand side, which users can utilize to create a study cohort, make selections to answer their clinically relevant questions. For example, today, I am interested in looking at the clinical profile for non-diabetic GADA-positive donors. So you can see that there are 17 donors that match these parameters out of 193. And on the right-hand side, you can see that these donor cards show you a snapshot of different information. For example, you can look at the autoantibody positivity profile for HPAP-008. This donor is GATA positive. You can look at the endocrine cell composition information and medical history. And if you're interested in looking at the imaging data for some of these individual donors, you would select that View HPAP ID button on top of that donor card, which will open this tab, and you can look at not just clinical data, but also like IMC data, as I'm showing you, histology data. And remember, I told you our imaging data is available on PanKbase. So if you want to jump over to their image viewer for this image, you can click on this button, this PanKbase image viewer button, and that will take you to PanKbase. On this islet composition page, you can also look at the endocrine cell composition information generated by CyTOF for this individual donor. And for some donors, we also have the immune cell percentage data. Now, perhaps you're interested in downloading the raw data that for this selected subset of donors. So as you can see on this page, the experiment data download page, which you can access using this drop down, these filters I had selected previously at the donor information page is still maintained. Even though I have pre-selected them today, in reality, that's what happens. These filter selections stay maintained throughout different data download options. So here, I'm interested in downloading some specific assay data for non-diabetic gather-positive individuals. On the right, you can see this matrix where on top, x-axis, all of the assay names are listed. And then on the y-axis, the cell and tissue type of origin for all of these different assays are listed. For example, I am interested in downloading the islet perfusion data and single-cell RNA-seq data for non-diabetic gather-positive individuals. So as soon as I select these assays from top over here, everything that's available for these donors, for these 17 total donors, they get highlighted in yellow. And you can see that I have selected download size of around 425 gigabytes, which is a lot. And as soon as these selections get highlighted, another box on top over here gets highlighted. This button over here allows you to download a bash script that packages all of the files that are part of this selection, and also has pre-selected parameters that allows you to connect to our servers and download everything you've selected. You can also edit this bash script per your need. And once you download using the script, once you run the script, it downloads two folders in your directory, a zip folder for the data, the 425 gigabytes of data, and a folder for the metadata, which is nothing but contextual information on the assays that you selected. It has information like what platform was used or what version of the software was used and so on. Perhaps you're interested now in a more granular breakdown of data availability for different individual donors. So for that, I have come to this another tab, by data status and donor tab. And here you can see a similar matrix. Again, on the left-hand side, notice that the selection filter selection is maintained. Every count over here represents the count for data availability for these 17 donors. So on this matrix over here, all the assays are again listed on top, but this time the data availability is shown by each donor ID. So for example, once again, I'm downloading islet perfusion data and single-cell RNA-seq data. As soon as I do that, every donor that has data for these two assays, they get highlighted in yellow. And any donor that does not have data for a specific assay does not get highlighted. And we label them as unavailable. And we even give you the reason for unavailability as you hover over the cubes. You can see over here that there are different colored boxes. These are nothing but labels that we use to communicate to the user exactly at what stage of data availability different data sets are for different assays and different donors. Remember what I told you earlier, that there are many reasons because of which there can be difference of data availability. For example, available means data that are currently available on PanK-DB for download. In progress means data sample has been procured for a specific donor and are being processed, and data will be available in the near future. Unavailable means that the data will never be available on PanK-DB for various reasons like experimental failure or lack of islet isolation, for example. And revoked means data that was previously available on PanK-DB but was removed by the data generation team. There are very few of such sequencing and this label, and they are mostly relevant to sequencing data. And we also provide you the reason behind both unavailable and revoked labels as you hover over the cubes. Okay, so now lastly, there is one last way of downloading data from PanK-DB. Once again, on this page, by donor list page, you can see that all of the 17 donors that match the parameters of my cohort selection are listed. Perhaps you're interested in downloading a specific file, for example, the flow cytometry files for B cells for HPAP24, and I'm interested in downloading one specific file. Perhaps in the previous download something was corrupted, and I just want to download this one file. So I navigate to the specific folder I'm interested in and the specific file. I make a selection and then download using this button over here, download selected. Please note that all of these different facets of data download are all part of the experiment data download page and are different tabs on this page. So you're not opening multiple windows. Okay, so up until here, I've shown you how to create a study cohort, how to query our data and download the raw data. But we have tools that allow you to make the decision on what data sets you can download or you should download, that helps you answer your clinically relevant questions. And those are our interactive analysis tools. These were developed by our team or were already existing, but we used our data and implemented them for our data set. So you can access these tools using in the interactive analysis page available in this dropdown and also on the home page. So once you open this page, you will see all of these different tools, collection of tools listed. For my use case, I'm going to start with this tool and then go up this list one by one. I'm not going over all of the tools in the interest of time. I'm just going over some of them, but I urge you to play around with our very impressive set of tools over here whenever you have time. So let's repeat my use case scenario today, that compared to the general population, GATA positive individuals are more likely to develop type 1 diabetes, but not all of them do. So I want to use a data set, an experimental data that can give me an acute information on islet function in different disease groups. And that is because that is why I'm using this islet function data portal. So as you select this button, it will open this portal, this islet perfusion data portal. Here off the bat, you can see a dynamic plot showing you minute by minute islet secretion information for insulin, which is in blue, and glucagon, which is in red. For our case here, we are only going to focus on insulin secretion. And these secretion profile are in response to different stimuli. For example, G3 stands for low glucose and G16.7 is high glucose stimuli. If you're interested in learning more about what this islet perfusion experiment is, how it was conducted for HPAP, why it's done, we have a wonderful tutorial on our tutorial page. Today, I'm only going over the higher level information. So on the left-hand side, once again, you can see that you have these demographic and medical history filters that allow you to create a cohort, which I have already done. I have selected a healthy age range of 20 to 30 years of age range. I've selected non-diabetic individuals, and I'm interested in GATA positive individuals. So I have made sure that they are not positive for any other type of autoantibodies. So you can see that per my selections, there are 10 donors that match this criteria. And all of these 10 donors get listed in this drop-down list over here. And out of these 10 donors, I have randomly selected 2 donors, HPAP 49 and 50, and because I've tagged them, and because I'm interested in coming back to these 2 donors and compare their secretion profile. So how do you tag these donors on this page? So here is the flag, here is the button which allows you to do that. So as you tag these specific donors from within your cohort, they get listed outside of the dropdown in a separate list. So you don't have to browse through the 10 or 20 donors of your cohort all the time, and you can just keep coming back to a narrower set of donors. Okay, let's look at the secretion profile for HPAP 49, a non-diabetic GATA positive individual who's 29 years of age. If you look at the insulin release profile of this donor, look at the y-axis over here. Looks like this donor has a pretty healthy profile for insulin release with at least 10 nanogram per 100 islets. Now, let's look at where this donor's profile lies compared to the rest of the HPAP cohort, because this page allows you to look at a cumulative distribution plot that would answer exactly that question. So explanation of exactly what values were plotted here and how these values are calculated are in that tutorial, and in the interest of time, I'm unable to go over the details of that. So if you scroll down, you can see that for HPAP 49 with a healthy-ish insulin release profile, it falls somewhere in between in relation to the rest of the HPAP cohort. Its insulin release is more than 75% of the HPAP cohort. So it looks pretty healthy. Now let's look at the insulin release profile for HPAP 50. So you can see HPAP 50, age at 21, non-diabetic GADA positive donor. The insulin release seems pretty low. According to this y-axis data over here, it's less than 1 nanogram per 100 islet. It's pretty poor, and if you look at its profile compared to the rest of the HPAP cohort, it falls in a very lower percentile, where more than 95% of the HPAP cohort has higher insulin release than this donor. So I think it's safe to say that HPAP 49 with a more healthy insulin release is behaving more like a healthy donor, clearly, and HPAP 50 with very low insulin release is behaving more like a type 1 diabetes donor, although they are clinically very identical, non-diabetic GADA positive in the same age range. Now, as you know, in type 1 diabetes, the beta cell count that releases insulin, that count is extremely low in type 1 diabetes versus healthy donors. So I'm interested in seeing if the beta cell count can also show this difference in insulin secretion, or can explain this difference that we just saw here. So for that, I have come to the donor information page, like I showed you earlier, available on this drop down. And if you look at the donor cards for these two donors of our interest, and focus on their islet cell composition profile, the beta cell count for these two donors is almost identical. So clearly, this data does not explain the physiological difference we saw earlier. So perhaps there is a genetic predisposition that these donors have that can explain this physiological difference. So for that, I have come to another portal available on PANC-DB. It's called the Genetic Risk Score portal. So here you're looking at the genetic risk for developing type 1 or type 2 diabetes, which can be quantified using a score plotted in this distribution plot. And these static lines basically show where an individual lies within this plot. So clearly for this individual donor, HPAP 49, which was behaving more like a healthy donor, has an extremely low risk of developing type 1 diabetes. And if we look at the genetic risk score for HPAP 50, which was behaving more like a type 1 diabetes patient, has an extremely high risk of developing type 1 diabetes, which is higher than 91% of the HPAP cohort. Now, I can clearly see that there is some difference that can be explained genetically. And if I dive deeper into the single cell RNA-seq data for these donors, and look at the insulin gene expression for these donors, which you can see in this hue map, where the neon green areas are little to no gene expression of insulin gene, and the non-green areas are the higher areas of higher expression. And here you can see histogram showing you the level of gene expression in beta cells for insulin gene, which is selected over here, which you can find search using this search bar. And you can see that HPAP 49, that was behaving more like a healthy donor, has extremely high insulin gene expression, versus HPAP 50, that was behaving more like a type 1 diabetes donor, has a low gene expression of insulin. So I hope, so this is the end of my live demonstration, and I hope I have been able to show you the utility of our tools. There are many more tools. Like I said, we have a genome browser that you can go through. But I would like to end by telling you quickly that we are currently working on a very interesting project where we are training an AI-based image analysis algorithm for our HNEN codex data, and seeing what they can detect, and if they can detect cell clusters. And maybe by the next year of D-Challenge, let's see if you can beat us to this interesting challenge. So now this is the end of my presentation. Thank you for having me. I'm sorry for going over the time. I thank Dr. Golnaz Wahedi and Dr. Babak Faryabi for leading the PANC-TB team, the PANC-DB team, and various HPAP collaborators. Thank you so much.
Thank you very much. So yeah, since we're over time, I will turn right over to Marcella, who will introduce her team.
Okay, well, thank you so much, Jeff. Pancreas Knowledge Base, or PanKbase, is a new resource of the Human Islet Research Network and the diabetes research ecosystem. I'm Marcelle Brissot. I'm one of the PIs of the Pancreas Knowledge Base, and it is my great pleasure to introduce 3 presenters from our team. They are working on different aspects of Pancreas Knowledge Base, we are gonna start with Dr. Ha Wu, who is a computational scientist based at University of Michigan. We will continue with Parul Kudarkar, who is a software engineer based at University of California, San Diego. And finally, we have Dr. Fan Feng, computational scientist based at Vanderbilt University Medical Center. And with that, I'm gonna turn it over to Ha. Thank you.
You very much. Yeah, very excited to be here today and talk to you about PanKbase. And as you can see from the HPAP presentation, we're in such an exciting time that we have never had so much data. There are many different initiatives out there currently trying to address different phenotypes and different challenges in the biology space. Especially with the pancreatic specific efforts, we are part of HERN, which is the Human Islet Network, a research network, and we also collaborate with HPAP, IDP, and NPOT to have different data modalities in PanKbase. But as we all know, with more data sets, there are more challenges coming across different data ecosystem. One of them is that the data right now is quite decentralized. So they are spread across different resources, different repositories, and it could be really hard to find out which data set you can use. Secondly, some of them can be inaccessible since some of the data resources are catering for different groups of users. So the way that certain data is presented could be inaccessible. And lastly, some of these data sets are non-standardized. So they have different representation in terms of metadata, the data format, and it makes it really difficult for computational scientists to access the data and understand what's going on. So given all of that, we, as PanKbase, would like to come in and help solve some of these challenges. So in my part of the presentation, I will show you some of our effort in collecting and building this comprehensive collection of islet single-cell data that is a really highly demand comprehensive map that the community has expressed to PanKbase. So in our comprehensive single-cell map, we have data from 3 different tissue sources and 200 samples from 10 different studies. And as you can see on the screen, we have citation for all of the studies that we have collected data from. So this is very exciting, but when data is not properly integrated, it can also introduce a lot of challenges. For example, the data may cluster based on technical variation. As you can see, some of these clusters are dominant by one chemistry, or sometimes they may cluster based on tissue or data sources on the second hue map here. And also, if the data is not properly QC or quality control, some of these samples may exhibit ambient RNA or doublet problems. And therefore, with that, we would like to process the data in a very systematic way in PanKbase. So on the screen, I'm showing you a very big pipeline that we employ in PanKbase. So briefly, we started out with RNA-seq raw reads, and we carry out multiple steps of quality control for both read level and barcode level. And uniquely, we also carry out donor identity checks so that we can map the samples we have in the map with all of the rich metadata that we have collected in PanKbase, which Parul will touch on in the later part of the presentation. After this, we carry out RNA ambient removal, and then we collected multiple quality control plots to decide what data is high quality to go into the single cell map. And in the presentation, I will touch on a couple of slides that I'm showing you here. And lastly, we go through doublet removal before integration, and then we have had the chance to carry out some of the downstream analysis looking at differential expression analysis. On the slide, you can see there's a QR code that will lead you to the GitHub page of PanKbase, so you can explore all of the code that we use in PanKbase. But if you're interested in looking at the exact code that goes into the single cell map, you can look at the directory PanKbase single cell RNA-seq. So as promised, we'll go through one of the QC plots that we used in PanKbase. Then that one is the barcode rank plots, which is a very useful tool to decide the quality of a single cell RNA-seq library. On the screen right now, I'm showing you a barcode rank plot of a typical library where the x-axis is the ranks of the barcodes and the y-axis is the number of UMIs. So a barcode with a higher UMI will have lower ranks. So as you can see, we can appreciate that this typical library has two distinct knees and a very stiff cliff between the two knees, signifying the transition between groups of high-quality cells and low-quality cells. However, in a library with loss of single-cell behavior, we only see one knee and one stiff cliff and no second knee. And in a library with heterogeneous behaviors, there are multiple knees showing here, and then the changes between the knees are quite gradual. So in order to use this barcode rank plot and ensure that the interpretation of different analysts of the same plot is consistent, we set out to quantify the knee numbers on a barcode rank plot. And what we do is that we can calculate the degree of changes across the barcode ranks and infer the number of knees. And what it looks like is that we would plot the ranks of the barcodes as normal, but then on the y-axis, we plot the discrete differences between two executive barcodes. And in the peaks that I'm marking here, they correspond to where the barcodes changes very drastically, and that corresponds to the two knees that you can see in a normal barcode rank plot. And the number of knees in this typical library is 2. In a library with loss of single cell behavior, the number of knees is only 1. In a library with heterogeneous behaviors, we see that the number of knees is 3. So by having this quantification, we can make recommendation about which libraries need further inspections, and also we can ensure the reproducibility of the analysis. So all of this is just one very quick snapshot into the many steps of quality control that we have carried out in PanKbase. Currently, the integrated single-cell map of isolated cells are available on PanKbase. We have 448,000 cells across almost 200 samples and 141 donors from 10 different studies. We identify the 13 major cell types in an isolated islet. And as you can see here, the number of cells, samples, and donors are very well distributed across different tissue sources or data sources. And we hope that these numbers will keep on increasing as more wonderful resources as HPAP keep on producing new data. And if we plot the technical variations such as chemistry and tissue or data sources, you can see that the technical variations are now well integrated. So the PanKbase integrated single cell map is now publicly available on PanKbase, which you can access through this URI. The intermediate files such as count matrix or read alignments are also available through the data library that Parul will talk about in the next couple of slides. And if you're interested in exploring the downstream analysis that we have had the chance to work on, the differential expression analysis, between T1D and control samples, you can look at the differential expression gene browser, also available on PanKbase. And with that, I'm gonna hand it over to Parul to talk to you about the data library.
Hello, everyone. So I'll be talking about the data library, which is one of the component of PanKbase. You can access the data library at data.pankbase.org. The mission of the data library is to provide deeply curated, high-quality data sets from human islets in computable form, following the FAIR principles, which is findable, accessible, and interoperable, as well as reusable. We have aggregated data from key resources such as IITP, Protolabs, HPAP, NPOD, and as well as ATI. So the data library contains 6 interconnected data types, right from the human donors all the way through the final analysis results. Everything is linked through standardized identifiers. So you can trace from donors through biosamples to experimental assays, as well as the analysis results generated from these assays. So what can you find in the data library? So the data library has harmonized metadata such as human donor, biosample that are aggregated from IITP, HPAP, ATI, and NPOD, experimental assays generated from these donors. So at the moment, we have single cell RNA-seq and single nuclear ATAC-seq. Then we have process data, which you can find under the intermediate analysis results. And these use standardized pipelines and then generate underlying intermediate results such as sequence alignment files, count files from these samples. Next, we have analytical resources and results that you can find under principal analysis card. So these are results which include integrated single cell map that Ha just mentioned. We also have donor by gene count matrices and results derived from these resources, such as differential gene expression results. And finally, we also provide detailed reporting, which is the underlying schema for all these data types that generate the metadata, as well as the data processing standards used by the PanKbase team and the pipelines underlying them. So how can you access the data in the data library? So there are 3 key ways you can access the data. You can search the data. You can access the data by browsing the data card that's on the home page. The next strategy is you can filter the data by key data attributes. And finally, you can use free text on the home page search bar. You can search for disease term, technical term, or sample type. One key advice that I would give is the term that you are searching for, put it in the quotes for exact search results. So how can you access the information that's in the data library? There are, again, 3 ways you can access the data. So you can access the data directly through the web interface. Then you can use, we have created Jupyter scripts, Python scripts on the Jupyter notebook. And you can, so these are the common workflows, and you can access the metadata files and the underlying data files via the REST API on these scripts. And finally, for those who are computer science enthusiasts, you can use the REST API. And next, I will hand over to Fawn, who will be presenting on one of the pillar of PanKbase, which is Pank. So over to you, Fawn.
Okay, thank you so much. I'll get started. So as Ha and Peru introduced, PanKbase is a centralized knowledge base for pancreatic and islet research. So actually, after collecting and carefully quality controlling those data, our goal is not just to list all the data sets on our website. We want to truly integrate them in one unified framework. Therefore, we develop a knowledge graph database, namely PanKgraph, in the PanKbase system for deep data integration and cross-model exploration. So here's one schematic figure showing how PanKgraph, the knowledge graph, looks like. In PanKgraph, each node represents an entity, and they can be genes, they can be variants, they can be the abstract concepts such as gene functions. Each entity carries properties for the details of that entity, and all the entities they are connected with edges, which represent relationship derived from different data sources. For example, if we check this relationship, it is from PanKbase single-cell RNA-seq data, which are introduced. It demonstrates gene expression and differential gene expression. And this edge is from our fine mapped GWAS data, representing the genetic association between this variant and type 1 diabetes. So with this knowledge graph, we are trying to integrate biomedical knowledge from different resources in a unified framework. Currently, PanKgraph covers genes, genetic variants. We have fine mapped QTLs, or quantitative trait loci, of human pancreas and islets. We have GWAS of type 1 diabetes and differential gene expression from PanK-based RNA-seq data. We also cover some selected literature from Human Islet Research Network. Another advantage of this knowledge graph framework is it also links back to the original data sources and provide data provenance, which ensures the transparency of our work. For example, if we query the gene CFTR, we'll provide the detail of that gene, which is actually from Ensemble, so we'll refer back to Ensemble. We will provide the relevant publications, which links back to PubMed. We also connect to other resources like GTEx, like gene ontology, and definitely will connect to the internal PanKbase resources like single-cell browser and data library, which were introduced by Ha and Peru. We believe that by seamlessly integrating those data modalities together, PanKbase can move from data to knowledge. Here we developed a web interface for users to access PanKgraph. We have some example questions you can ask on this website. For example, you can query if a gene expression is different between type 1 diabetes and healthy samples. And you can also query the QTLs, quantitative trait loci, in human pancreas and islets by providing the gene or the variant you are interested in. I'll use this as one example. If you query the gene GAS2 and ask which variants are fine mapped QTLs, it will tell you we find the eQTLs, expression QTLs of this gene in human islets. And if you click this download button, you can get the tabular data of that variant, that credible sets. And if you click this view, it will jump to a page for the details. On the detail page, you can check the graph query result. If you hover on one of the nodes or one of the relationships, it will show the details of that node, that relationship, along with the reference, the provenance back to the original data source. And we also provide the interpretation with large language model to interpret what this result means. And as we introduce, we have data provenance. We'll refer to the publications which are related to this query. And if you want to understand like how this eQTL is calculated, how we performed fine mapping, here are the links to our internal resources about the pipeline and the details of those data sources. Currently, this website only supports template-based questions, but we are working on a more flexible version that allows users to ask questions in natural language. That will be powered by our PanKbase AI agent, which first translate your question into queries and then return and interpret the query result. That will be ready pretty soon, which is very exciting. And I also understand a lot of the audience are computational biologists. So if you prefer to access this resource with code or plan to submit batch queries, like you want to submit like 1,000 queries at the same time, we also provide an API. You can find the documentation in this page. Here you can also find the schema which are the metadata summary of PanGraph, which help you write valid queries. So as a summary, type 1 diabetes is a very complex disease. First, it is a polygenic disease with HLA playing an important role. And then the effect of genetic variants, they percolate through layers of complexity, epigenome, transcriptome, proteome, cell and tissues, and their physiology, all until they influence the final trait of type 1 diabetes. What makes this more complicated is although the genotype is invariant in one individual, all the other layers, they are also related to environment, age, sex, and a lot of other factors. So because it's complexity, we need to integrate data from all those layers. We need the combination of basic science, clinical science to understand type 1 diabetes. Therefore, one of our major plan in PanKbase is to expanding it by incorporating more data modalities, and we'll follow the same strict and systematic workflow, just like we introduced for single-cell RNA-seq. We'll use that workflow to do like metadata harmonization, to carefully quality control, to integration with existing data sets. Through the effort of PanKbase, our aim is to uncover the molecular mechanism of disease pathophysiology and ultimately to advance disease diagnosis and prevention. So I would like to acknowledge the amazing team behind this work. Here are our PIs whose vision and leadership drive the PanKbase project, and the trainees who have contributed a lot to data curation, analysis, and platform development. We also want to express our deepest gratitude to the organ donors and their families. Without them, none of this research would be possible. So yeah, thank you for your attention. I think how many time do we have? We're open for questions right now.
Yes, you have 5 minutes for questions. So is there anyone on the call who would like to ask a question for the PanKbase group?
I think there's a question in the chat asking what mechanism you used at the back end of PanGraph.
Yes.
Yeah, it is Neo4j, and we deployed that on AWS.
Okay. Can I follow up the question? So
Yes.
I'm already very excited when I see this knowledge graph data. So if we are able to do the API query, it'll be very, very nice because at least I'm building the application. So I do need to like using API to access without having to download the data, right? So currently, I just did a quick look of the API, the documentation. So currently, we have to like basically write Neo4j Cypher query and then
Yeah,
embed
exactly.
it.
Yes, because
Okay.
it's based on Neo4j. So currently we support Cypher queries. That's why it's very important to like know which node types, which relation types are available. That's why we
Yeah.
also provide this page to refer to when you write that Cypher query.
Okay, great.
Yeah, thank you for the question.
Thank you. Very excellent.
Okay. I actually had a quick question, sort of in terms of sort of the integration between sort of the PANC-DB and the PanKbase knowledge graph. So how quickly, or what sort of the turnaround time between sort of synchronization of this data into the knowledge graph?
I think it's an excellent question. So I think right now we don't have a clear timeline, but we do have plans to incorporate more data, that especially the ones that are newly available through HPAP. So the challenge right now is that the quality control step is definitely quite tricky, and we want to be as rigorous as possible. And we've made all of the code as streamlined in pipeline via Nextflow. And we currently, I lead an effort to do like automatic thresholding, so you can minimize the data idle time as much as possible. So I think one challenge right now that is like an open challenge for everyone is to develop an AI that we can just feed in the data, and then we use the code and all the knowledge that we already have to quickly process the data and integrate it in the current map. And that's something that I really want to do, but I'm only one person. So the D challenge and these boot camps are excellent so that we can get more efforts in.
Thank you.
Yeah, exactly. And the multimodal data is very exciting, I think. I want to ask one more question about the eye receptor data. I don't know if you guys have also integrate the eye receptor data, because we are also introduced last week about the air data common. So I think it's also very good data sources that we want to use. I'm just wondering if there's any data entries into the knowledge graph, like can collect both data sets. That would be awesome if we can.
Yeah, I think as Fan shown, we wanted to incorporate as much data as possible. So it will be very helpful for us to hear your opinion that what data types are necessary. Currently, we spend a lot of efforts in introducing functional data into the knowledge graph. So all the parasitic fusion data would be available through the knowledge graph soon. But of course, all the receptor data would be interesting too.
Well, from iReceptor, I know there's some HPAP data in the iReceptor, in the Air Data Commons. So that would seem to be the same data, I guess. So you could integrate that way. I don't know the details, but that would be my
Is there any tech support for us if we have any questions or problems when we work on the project? Because
Yeah,
I think it's very likely that we have
yeah, tech support equals Brian Corey. So there's I think it's support@ireceptor.org or something like that. Yeah. But
Brian is tech support for the eye receptor, right?
for iReceptor, yeah.
I mean, for this PanKbase or the knowledge graph, is anybody can like help us doing the?
So I'll paste the email. So we have our email help@pankbase.org. I'll paste it in the chat.
There is also a community forum on our GitHub page for questions as well that get funneled down to the different working groups.
And this is Amanda, wonderful Amanda Huber, who just jumped to answer the questions. She's one of our project managers for Pancreas Knowledge Base. And
Nice.
she is the one who is funneling questions that through our general email that Parul just posted into the chat.
Thank you, that's awesome. Because we have very limited time period, so I hope that now questions can be prioritized.
Yes, yes.
Great.
Thank you so much. Thank you so much for those questions.
Thank you for the effort. It's very nice.
Okay, well, we are now at the top of the hour, so I wanted to thank everyone, especially our speakers today, for their great presentations on two, very high-value data sets and resources. And so, I'll let everyone go. Monica will be making these available through Sugar Science, and so you'll be able to see these online as well down the road. So hope everyone has a great day and a great weekend.